Okay, recording is on. Thank you for joining us for the Vaccine and Variant Education webinar. We know this topic is so needed in our country and all over the world, and we appreciate everyone joining us tonight and taking the time to educate yourselves on COVID-19 vaccines and their variants. I know there's a lot of questions and uncertainty at this time. My name is Katie Stevens. I'm the Executive Director of Team Telomere. My job is to support the mission of Team Telomere, which is to educate and make treatments accessible to patients with telomere biology disorders, and to make sure that we are educating the general public on how to keep those with telomere biology disorders safe. Joining us tonight is Dr. Sharon Savage, and I am privileged to have worked with Dr. Savage for almost a decade now. Doesn't seem like it can be that long, but in a professional manner, the last 5 years. So I will let her introduce herself and take over from here.
Okay, sounds good. I'm going to share my PowerPoint here. And there, you all see that pretty well. So I actually, I think I know everyone who's currently on the call, which is great. But just in case, or for those who might listen later, as Katie said, I'm Sharon Savage. I'm a board certified pediatrician and a board certified pediatric hematologist oncologist, and I've been working in the world of telomere biology disorders for over 15 years now, and have been focused on pediatric health issues for the entirety of my career. And for those of you who do know me, you'll know that I'm very much a physician and a scientist. And I think my approach to the pandemic and to helping everyone understand it is that we're really all learning about COVID-19 together. This is really an unprecedented time in that we have a new illness, a new virus. No one really knew what was going on, what to expect, and that happens at all levels. So I like to put this up as a reminder from some of our science classes in middle and high school, in that the scientific method is really critical in understanding not just the pandemic, but also kind of analyzing information in general. So the first part of the scientific method is the question. What do you want to learn? What question do you want to answer? You need to research it. There's a variety of different ways to do that. Hypothesize, kind of what do you think will happen based on your research? You do the experiments to test that hypothesis, analyze the data, and share it. And this is really what's been happening in real time throughout the last year and a half. And it's something I think that most of the public has not seen the scientific process happen in such granularity as we've seen it over the last year and a half. So you all know about genetics, but just as a brief refresher, because I'm going to give you some background on viruses, I think it helps to put it all in context. So our DNA is inside our cells, and we think of our DNA as the whole book. It's our genome. It has all the instructions for our bodies to do everything they need to do. A chapter equivalent in a book is a chromosome, and we have 23 pairs of those in our human DNA. And then the sentence is the gene. So in this example, which we use a lot for genetic counseling, the sentence is the gray cat ran down the hall. But if you have a variant in one of those nucleotides, in one of those DNA bases, or in this example, a variant in that the H was changed to a B, the sentence changes significantly and doesn't make quite as much sense. And so changes in DNA and in RNA, which we'll talk about, are part of DNA replication. It's a normal part of life, and it's something kind of similar to how we talk about genetic counseling and genetics for people with telomere biology disorders. Similar things actually happen on the viral level. So I've just talked about DNA, which is these 4 A, T, Gs, and Cs, we call them bases. So our cells have the DNA, that book that has all the instructions. Well, how do you get those instructions out to actually do something? That happens through a process called transcription, in which there are cellular processes that take the DNA as a template and they transcribe it, just like you could transcribe the sentence in that book, into RNA. And then what happens is messenger RNA uses this information in this transcription to then make proteins. So and messenger RNAs do this gene by gene in our cells. And the other key thing to understand is that messenger RNA degrades very, very rapidly. We say in the lab, DNA, you can isolate it from samples of mammals thousands and thousands of years ago. It's really stable. It's really hard to disrupt DNA in the laboratory, in the test tube. Messenger RNA, though, we say in the lab that if you look at it wrong, it degrades. It's very sensitive. It's very challenging to work with. And that's because it's such a valuable, important thing for the cell, that the cell doesn't want to have too much or too little. So it's tightly regulated. So viruses, what's a virus? The official, it's an infectious agent that contains DNA or RNA and a protein coat. It has no ways to move around. It has to be taken into cells to survive. And when it does, it basically hijacks the cell and uses the cell's RNA or DNA replication machinery to make more of itself. And then it has that cell release more of its replicated virus, usually because the cell dies after it kind of reaches the maximum ability. And there's evidence that viruses have existed in humans for at least 7 million years, so they're nothing new to us. There are a lot of different types of viruses and subtypes. There's even viruses that cause diseases in plants. This is an example of a specific RNA virus called the tobacco mosaic virus, which infects plants. There are viruses that just have DNA in them. There are viruses that have RNA in them. And then there are other types of viruses that are even more complex and have this complex structure that has DNA. But we're going to focus on this virus, the RNA types of viruses, because that's the type of virus that coronavirus is. So a coronavirus is actually a whole bunch of different types of virus. So it's a large virus that has a lipid, a fatty layer studded with proteins. So that's why you see those pictures like this all the time. And it infects birds, mammals, including humans. And coronaviruses have been the causative agent of other respiratory illness outbreaks prior to COVID-19. So SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2. And I want to remind everyone that this was a newly discovered virus. Even 2 years ago, we didn't know about the SARS-CoV-2 virus. And consequently, it was the cause of a new outbreak in late 2019 in Wuhan, China. So COVID-19 is the disease caused by the SARS-CoV-2 virus. And remember, this is a new illness, a new virus. So a year and a half ago, 2 years ago, before all this started, we knew a lot about coronaviruses. We knew a lot about respiratory illnesses. We just didn't have the specifics on SARS-CoV-2. So I want to also show you some data that I think puts it all in perspective and why, COVID-19 is so bad and why we're going to talk about vaccines in a moment. This is from a a mortality tracker that my colleagues here at the NCI developed very early on in the pandemic. And it's basically a way to look at causes of death in different populations. So what this is it uses updated data from the CDC. And if you go to that tracker, you can play with the settings and look at different things by different states and timeframes. This is a curve starting from March of 2020 up. This goes up through till almost June of this year. And you can see that this is the number of deaths per week due to COVID-19. It ranges from 3 to about 22,000 deaths in the country per week. When you compare that with number of deaths per week due to cancer, you see that from over the same time frame, the deaths from cancer are relatively stable, roughly 11,000 per week. This little tail here at the end is because the data aren't yet complete. You look at heart disease, death due to diseases of the heart. You see some ups and downs. This we do see a seasonal change sometimes in heart disease over the winters. But again, it's a relatively, doesn't change a lot. You see the range is like 11 to 14, 15,000 people per week. What I think is really striking is that when you overlay all of these different causes of death, and I'm just pointing out here, orange is COVID-19, heart disease is pink, cancer is this purple line. And what this shows is that basically all these other causes of death since March of 2020 have remained essentially the same. And we see this huge spikes as you see reported in the deaths due to COVID-19. So this is really, it's really disheartening news. And at the same time, it's something that it's nothing new, right? You all know that COVID-19 is bad, and the pandemic is really been going on for a long time. So what do we do? How do we end a pandemic? And in this case, I come back to Benjamin Franklin, who first coined the phrase, "An ounce of prevention is worth a pound of cure." And in making this, I learned that he actually said that in talking in Philadelphia with potential firefighters, and basically talking to them about how the importance of preventing fires is, because it's much better to prevent a fire than it is to have to fight a fire. It's the same thing here. It's much better to prevent a severe viral illness than it is to not. So how do we end the pandemic? Some of you know Dr. Neelam Giri here at the NCI. She's one of my colleagues. She was also the fourth person at the NIH to get the Moderna vaccine at the end of December. She also was a selfless volunteer in helping to test NIH staff for COVID and in vaccinating staff. Vaccines are really how we end this. The other way we end this is with masks and social distancing, which I know we're all very tired of hearing about, but this is the only way it's going to happen. So vaccines, how do vaccines work? And what they do is, and how are they developed? So I think of the vaccines very much just like in these images. You have a disease-causing organism. In this case, it's the SARS-CoV-2 virus. What happens is the scientists take a little tiny piece of that virus. In this example, and in COVID, it's the spike proteins. And they basically put it together with some stabilizers and sometimes preservatives, sometimes not, in a liquid. And they create this liquid that we've seen in the vaccines that is used to induce a vaccine or an immune response. Now, vaccines are also required to undergo the same exact types of clinical trials of testing as any other drug, any medication, anything. And this is what a lot of you have also heard about in the press as the vaccines were developed. There are early phase 1 trials where you have a small number of people and you're really looking to find the best dose with the fewest side effects. Phase 2, you expand and you do additional tests, including looking at efficacy. And then you expand in phase 3. So you gradually increase the number of people you're studying. And then you continue to monitor once it's in broader use. So we've heard a lot over the last year as well about vaccine efficacy and effectiveness. They're somewhat different, and it's important. So vaccine efficacy refers to how a vaccine performs in ideal conditions in a controlled clinical trial, whereas the effectiveness refers to how it performs in a wider population. So in this example, if you have the placebo group shown in blue, the vaccinated group shown here, and the efficacy is basically calculated based on the number of illnesses that are prevented in the vaccinated group compared with the placebo group. So a couple of things we talk about, and actually the efficacy of the COVID-19 vaccine is greater than 90–95%. In this example, if a vaccine has an efficacy of 80%, it doesn't mean that the vaccine only works 80% of the time. That's not the case. What it does mean is that in a vaccinated population of people, 80% fewer people will get the disease if they come in contact with the virus. Okay, so it's not based on per person, it's based on the population. And so in this example, everyone in this picture had the vaccine. They were all exposed to virus. Only 2 of them out of the 10 got the illness. That's vaccine efficacy. The fact that we're well over 90–95% for the COVID-19 vaccines is absolutely amazing and really encouraging. I'm not going to go into this in detail, just to say there are a lot of different types of vaccines. Some have been around forever. For example, smallpox was inactivated, and polio were whole inactivated viruses. There's other vaccines that basically have a very much mild virus that cannot cause severe disease. But the whole idea is to basically have your body make an immune response to help you have better ability to fight off an infection. So we've also heard a lot about mRNA vaccines because the COVID-19 vaccines are actually the first widely used mRNA vaccines. If you remember, I told you earlier, messenger RNA, mRNA is a very, very delicate entity, and it's degraded by normal cellular processes. There's absolutely no risk that the mRNA vaccines could cause infection or insert into your genome and cause mutations. That's absolutely impossible from the biology perspective. Can't happen. And there's also been decades of studies of mRNA vaccines that just haven't been pushed forward as quickly as we've heard over, again, the last year and a half. So how does the COVID-19 mRNA vaccine work? And that's where I'm focusing mostly, which are the Moderna and the Pfizer vaccines. So what happens is the mRNA vaccine, this actually says a lot of the same thing, and I have a figure that I'm going to show, but basically the mRNA kind of tricks the cell to make a little tiny piece of the protein that's on the outside of the virus. And then your body says, hey, that protein isn't normal. That's not part of me. I need to get rid of it. And so your body reacts and says, I'm going to make an immune response. I'm going to get rid of that little bit of protein. And when it does that, your immune system then actually remembers that process. And so here's another picture of this idea, in that you have a vaccine that has this mRNA that just codes for a small part of that spike protein. So just a little bit of it, that it basically goes into the cells, makes the spike protein. The mRNA goes away very, very quickly. The spike protein is then on the cells, and your immune system says, whoa, wait a minute, that's not supposed to be there. And what happens is you make antibodies, and then your body remembers that response, so that if it sees the COVID-19 viruses again, it can make a good response and move on. I'm going to skip this part because we've talked about this, but I'll share this link. It's a nice little video from the WHO of vaccine development and how things have moved forward. But I want to emphasize again that a vaccine protects an individual, but community vaccination protects the whole community, even those who can't be vaccinated. That's a real key part as well, especially as we're still waiting on the data for the trials for our children. So this brings me to viral variants, which are a big concern now. And I have a couple of great videos from the WHO that are very short that I think are good at illustrating why this happens. And basically what happens is that the more virus you have in a population, the more infections you have in a population, the more likely that virus is to mutate. And it's always going to mutate into what allows it to keep spreading and growing, right? That's where that's the preferred. So we hear about the Delta variant. What happens is the Delta variant that's circulating now is very much more infectious. And that's how it's able to sort of overtake everything, because the viruses are all about self-preservation and replication. So I'm going to show this video that I think is a nice way to illustrate this. So basically, inside a virus, we've talked about there are either RNA or DNA, and we think of these as a code. And if you imagine a bike lock like this, where you have 4 different options to code, and then if you imagine that the lock has 30,000 wheels, that's the genome sequence of an entire virus. And each time the virus spreads, it has to copy this sequence over and over and over again. But every now and then, as it copies, the wheels in that sequence can slip. In this case, you get a U changing to a G. Most of the time, these are tiny variations and they don't change how the virus works. Sometimes actually it's a good thing, because some of those variations don't work and it might make the virus weaker and these strains tend to die out. But sometimes the variations can make it stronger, like the Delta variant, better at transmitting, more resistant to the immune system, and then these strains can really thrive and take over everything. The good news with COVID-19 is that all of the vaccines that are currently approved work against these new variants. So that's really good news, and another reason to keep everyone getting vaccinated. So just another image of what causes a virus. Oops, next one, sorry. So how do we stop the spread of viral variants? This is really by continuing to be persistent and do what we do. And do what we've been doing. And this is another way to look at this, in that the more the virus spreads, the more variants are likely to appear. And we never know if a particularly strong variant is going to emerge, like this red one, like Delta. But we know that we can prevent it. So if someone wears a mask, if you stay home, if you're sick, you wash your hands, you get vaccinated, you can really change that entire timeline. And so really, I think where the pandemic is now is we need to change the timeline. We need to get back to the basics and prevent transmission of the infection. And I have one more short video. Oops, that's the same one, sorry. Here we go. And it's really a group effort. It's not something that one city, one state, one country can do. It's a worldwide effort to stop viral variants. And it's important because if you imagine if Area 1 is able to completely eliminate the virus, that's wonderful. But if area 2 doesn't, you run the risk of stronger strains evolving and eventually coming back to this area 1 and causing this cyclical issue. So we're only safe when everyone is safe. And this is why, you know, I'm really happy to be here and to talk about the vaccine and answer questions about the virus. So another reason why people should get vaccinated, and I think this came up in one of the questions that Katie received prior to our meeting, was an adult saying that, well, my kids can't get vaccinated, so, you know, there's no point in me getting vaccinated. The reason a parent should get vaccinated is to be alive for their children. So this was a study that came out just a couple of months ago in JAMA Pediatrics, the Journal of American Medical Association. There's been some press release on it, but the summary is they looked at the estimated number of children ages 0 to 17 who could lose a parent due to COVID-19. They did a lot of evaluations of existing data and complex computational simulations, but basically the number of children experiencing a parent dying of COVID-19 is estimated to be at least 37 to 43,000 children. And these are most of these, I would guess, were preventable or definitely are preventable with vaccine now. So the COVID-19 trials for children under 12 are still underway. And so now for children, we really are still doing our best with hand hygiene, fresh air, masks, social distancing. Now, I realize this isn't always possible, and I'll share some resources for school as well. But in my opinion, if you have a child who's 12 and over, they should get the Pfizer vaccine immediately if they haven't already. Everyone over 18 is eligible for Moderna or Pfizer as well. So a couple special topics, because I know this is an important topic for the community, as immunocompromised people. This is from the CDC website, and they just came out. Again, science is literally changing before our eyes. August 13th, 2021, that people who are moderately or severely immunocompromised are recommended to get another dose of either the Pfizer or Moderna vaccine as a booster. Now, who's immunocompromised? We've heard a lot about that in the press. People who are on treatment for cancers, people who have gotten an organ transplant and are taking medication to suppress the immune system, people who received a stem cell or a bone marrow transplant within the last 2 years, or if you're taking medicine to suppress the immune system. There are certain inherited disorders that are associated with an immunodeficiency, people who have advanced or untreated HIV infection, people who are on high-dose corticosteroids, that's prednisone usually, or other drugs that might suppress your immune response. Now, everyone is different, and so the most important thing that people can do is talk to your doctor about when it is the right time for an immunocompromised person to get vaccinated. So a couple of questions that come up is, so I'm immunocompromised. When can I get my 3rd dose? I really want to get protected. It's recommended right now that it's at least 4 weeks after the 2nd dose. Can you mix and match Pfizer or Moderna vaccines? And the answer is no. The current recommendation is to use the same vaccine. Now, what about the Johnson & Johnson, the Janssen single vaccine? They're studying that right now. The preliminary data from what I've read are promising, but there aren't enough data yet to recommend whether the people who are immunocompromised who got the J&J vaccine need a booster or not. So why doesn't the vaccine work in immunocompromised people? the whole thing with being immunocompromised is that your immune system just doesn't work as well as it should. And that's why you don't necessarily make a normal response to any kind of vaccine, not just the COVID-19 vaccine. And then a really important question is, what are the risks of a third dose? So far, there are still more studies going on that my colleagues and many others around the world are looking at the vaccine responses in people who are immunocompromised. But so far, the data are reported that after the third dose, the side effects are about the same as after the 2 doses. Fatigue, pain at the injection site were most common, and overall the symptoms were mild or moderate. So I'm not going into this in detail because it's a huge topic, but I do want to point out the challenges of kids in schools, which we're all dealing with in our own different ways. And healthychildren.org is a really great resource. It is run by the American Academy of Pediatrics, which is the leading child health advocacy, education, and medical clinical group in the world, really. They have a great resources at healthychildren.org. So I would encourage you all to check that out. And another, of course, resource is the CDC website, and they have a lot of details. Additionally, I've been really impressed that all of the state health departments have been working really hard and have really good guidance that's all scientifically based, data driven. And this is from the CDC, which I agree with, that transmission of SARS-CoV-2 within schools can be limited with strict implementation of layered mitigation strategies, including universal and correct use of masks and physical distancing. And then just as a couple things to point out, there's a lot of stuff out there that just is gotten out of control. Mythbusters are, I think, a really important piece of this whole puzzle in making sure that we're educated. And both the CDC and the WHO have great sites that are data-driven mythbusters. For example, the CDC, there was a lot of speculation. Do the COVID-19 vaccine contain microchips? No, absolutely not. And it says here exactly, you can go to see what the ingredients are, and talks about how the vaccines actually work. One from the WHO that also was a crazy idea, that basically said there are no data. It's actually physically impossible for viruses to travel on radio waves or mobile networks. And if you think about it, COVID-19 is spreading in countries that don't have 5G networks. So again, going through and educating ourselves to help educate each other, these are great resources, I think, too. And then I want to just point out, because in addition to the COVID-19 pandemic, I think we're also suffering from an infodemic, in that there is so much, the science has been moving so quickly related to COVID-19. The data are being generated so quickly. The scientists and clinicians are working very hard and compiling that data and then sharing that data. And because it's changing so quickly, I think that's where, in part, it has added uncertainty and added more speculation. And so I like this infographic, basically about helping. This can be on any topic, but assessing the source, going beyond the headlines, identify the author of this source, check the date. Is it relevant? In fact, especially in something changing so quickly, when did this happen? We did not have protective equipment a year and a half ago. Our guidance was a little bit different, but then we changed based on evolving data. Examine the additional supporting evidence. this is really important as well, that we all need to do. And I think in science, we really are making a conscious effort to check our biases, because we all think things, and really we're not here to give our thoughts. We're here to look at facts and data, and then going to fact checkers that you do trust, universities, scientists, physicians, etc. And then this is just some suggested reading I'll leave out. These are the website links. I've tried to put them all throughout the presentation, and I'll share some of the summaries with Katie as well. I thought this was a great book for kids. Bob explains how the COVID-19 vaccine works. And then these other two books I put for perspective. The Great Influenza was written a few years ago, and it's actually about the influenza pandemic in 1918. And it's a great historical read that also talks a lot about the early development of testing and vaccines and isolation, because at the time that was new as well. So it's an interesting perspective. You might not want to read it now, maybe later. And another one that I think for people who have questions about vaccines and are looking for data, this is a good summary by Dr. Paul Offit, who's at CHOP, Children's Hospital of Philadelphia, about what vaccines are, how they work, what they do, and how they truly save lives. So that's all I have. And if we have time, I'd be happy to take questions, or if anyone wants to go back to any slides, we can certainly do that as well.
Great, Khalil, if you just want to.
Sure. Hopefully I'm unmuted. Thank you so much again, Dr. Savage. That was very didactic and helpful to sort of go through the entire biology behind this. One of the things that has come up on a recurring basis has been sort of breakthrough cases and vaccine efficacy. So for those people who are immunocompromised and have 3 doses now, or in the last couple of weeks, you said August 13th, or those people who aren't immunocompromised and have antibodies from these mRNA options that we have, Moderna and Pfizer, what sort of behavioral changes does a breakthrough in sort of the traditional sense, right? Does it mean that vaccine efficacy is no longer what it was, or should we think about it in sort of a different context? And I say behavioral because I think a lot of us who are reading and getting information from Israel, from the UK, different information is circulating, and we're hearing it anecdotally of people getting COVID. You start scaling back and going back to sort of not even just mask wearing, but pulling back from daily activities, et cetera. So
yeah, so an important thing to remember is that the vaccine is not meant to prevent people from ever getting sick. The point of any vaccine is that, yes, there is a high efficacy and you can really prevent people from getting sick, but really the end goal is for people to not die from the disease. So the COVID-19 vaccine prevents people from dying from COVID-19. The breakthrough infections that happen are rare but happen, and but those are always mild to moderate at most in the healthy, otherwise healthy population. And one of the questions is, so for people who are immunocompromised, and a really important question is that how good is their antibody response to 1, 2, 3 doses? And each person is going to be different, and it's going to be dependent on their degree of immunocompromise and on the type of immune compromise they have. So that's where working with the home doctors is really important. There are some docs will monitor antibodies after vaccine and recommend another booster. Some of those are data that are still kind of being undertaken. One of the things that I think is also important to remember is that actually I didn't talk about this, but people who are immune compromised and who get COVID may actually have a higher viral load, meaning they have more virus in their body than someone who's not immune compromised. And that's because their immune system just doesn't work as well as it should. And so they could potentially pass it on to more people than someone who was not immune compromised walking around with COVID. As far as kind of prevention behaviors and things, I think that, we all should be wearing masks in public. I think I'm really worried about what the winter's going to bring as we start moving inside more. But I think, we need to get vaccinated. We need the vaccines to be reviewed, the data reviewed in the pediatric trials, and get the kids vaccinated. Does that sort of answer what you were thinking?
It does, yeah. The only sort of part I think that lingers is, particularly for this community here, when you think about what you were saying about the vaccine being a tool to make sure that those who get COVID don't ultimately die from COVID. But there are a lot of people, whether they be children or immunocompromised communities, including this audience, that you would want to make sure that you don't bring COVID home to them if you are one of the healthy people that can take the vaccine.
Right.
And so I think the breakthrough infection, it frustrates the effort ultimately, because prior to Delta, those of us who were vaccinated would venture out into the world, masked, but certainly taking precautions, but confident that if we were exposed, we would, with a high degree of certainty, 95%, 96%, not bring something home to someone who is immunocompromised. And now, in sort of this instance, now we're saying, even if you do everything right, our mask doesn't protect necessarily, us from getting infected. Ultimately, it's a conduit for us to not pass it along. So it almost sets us back, because when you're in a household with someone immunocompromised, you just can't afford the risk. At the end of the day, is what.
Yeah. So I hear what you're saying, and sort of where the question and the data comes from. So a very good thing is that even there's data out now that for healthy people who are vaccinated, if they get a breakthrough infection, which is rare, right? But it does happen. If they get a breakthrough infection, the amount of virus that they're carrying around is much, much, much lower than in someone who was not vaccinated. So, as a healthy person, doing all the right things, being vaccinated, wearing a mask in public, washing your hands, the chances of bringing it home to an immunocompromised person, I think, are really, really low. That said, nothing's ever 100%, but it's pretty close.
Thor?
Yeah. Thanks, Dr. Savage, for this. One question I'm thinking of when we come to booster shots is that if you were someone who had one of those rare side effects like pericarditis or whatever, would you expect that to happen again with the same with a booster from the same type of vaccine?
So that's a good question. And it's something that I know is still being studied, and I haven't looked at the data specifically, but in general, if you have and it also depends on the severity of the response. But in general, my first inclination is that yes, you would be at increased risk for some of those other more serious side effects. And those side effects are, of course, very, very rare, but things that need to be monitored for. And in fact, I think there are also some studies that are ongoing for people who had more severe side effects, who then, and thinking about getting boosters for everyone. And they're looking at that. My first reaction is that it's probably going to be more, it might be more of a problem. And that's where if anyone had a severe side effect, they really need to talk closely with their doctor and kind of see where the data are taking us when it's time for those boosters.
Jennifer?
I have the unique opportunity to be able to live in a community that really isn't buying into mask and all of that.
Yeah.
And I guess what I'm asking, is there been a study in any of the schools, because I'm also working the school system, is there been a study that I can look at or I can show the faculty or the staff that shows the spread rate with those that are not wearing masks right now versus those that are wearing masks? Because you would have thought the world was coming to an end when at the last minute our school board finally decided to go back to wearing masks, which I was very thankful for. But we're really struggling with this, and I'm just trying to find out if there's a study or something that I could read about to kind of get a better understanding of how to answer that question.
No, that's great. And yeah, there are, and I can get you some more information. I just don't have it at the top of my head. I know there have been a series of case studies and case reports of how masks prevent transmission in situations similar to a classroom and in classrooms.
Sharon, maybe after or in the next week, we could even put in our resources link, resources for COVID-19 and, proven studies, so that we can, those of us in the same position as Jennifer, trying to prove something, can utilize it from a resource like Team Telomere to take back to their communities.
Yeah, absolutely. In fact, I know who I can contact already, because I have a colleague who presented to their board of education for their county on exactly this thing. So I'll get some information from her.
Okay, thank you. Jen Chaney.
Hi, Dr. Savage. Thanks for doing this. Really appreciate it. I have an incredibly basic question prompted by one of your slides that was defining what immunocompromised means. And I'm just wondering, like, my son is in bone marrow failure, but his blood counts are not super low. If you're in bone marrow failure, does that make you automatically immunocompromised? Or, what are the factors that would define whether you are or are not?
So that's not a very basic question. It's actually a really complicated one. What's interesting to a certain extent, so there are, let me just say, so in thinking of, I'm going to get to the bone marrow failure part in a sec. When we think of immune compromise, there's actually different types of being immune compromised. There's one is having low neutrophils, which are the type of white blood cell that fight bacteria. Then there are also types of immune compromise caused by either abnormal function or too low numbers of lymphocytes. And lymphocytes fight viruses, but there are also different types of lymphocytes. So there are T cells, which basically, for the most part, contain the memory. So like, for example, if you get a vaccine and then you get exposed to that disease, the T cells are the ones that they're actually called memory T cells. They remember and they're like, oh, I know what to do. Let me go kill this virus. There's also, though, a B cell, which is a type of lymphocyte. B cells make those antibodies that we make when we get a vaccine or we get an illness. So there are different types of immune compromise across the whole spectrum. That said, though, people who have bone marrow failure and have basically, it depends on the disease, it depends on the a lot of things, but it depends on the specifics of the disease and of the genetic defect and a lot of different things. But overall, if the counts are pretty normal and it's someone with an inherited bone marrow failure syndrome, they should have a pretty normal immune response.
Okay.
It's when those counts start dropping that we start getting worried. But if the counts are pretty close to normal or a little bit low, the chances are pretty good that they're going to make a good immune response to anything they see.
Okay. Can I ask one other quick question? So in terms of getting the booster then, for people who like, how do you show that you're immunocompromised? Like, do you have to show some kind of proof of that to get the booster after 3 months, or do you, how does that work?
I think, that's probably going to be community dependent. I know here we're still kind of talking about how to roll it out. And I think that's where I would kind of go back to your community doctor, as far as how that is happening, because I think it's still a relatively new recommendation and some of the health departments are still kind of figuring it all out.
Okay.
Yeah.
All right. Thank you.
Thanks. Megan? You're muted.
Hello.
There you go.
I'm unmuted now. I'll turn on my video. Hi, everybody. My question was probably very similar to Jen's about the immunocompromised. So with Gabby, we often have people just assuming that she is immunocompromised, and, making sure we're keeping her safe.
I would do that for someone like Gabby.
Okay. That's where I'm sort of heading. Yeah, obviously we're 6 years post-transplant. Her blood's, I'm told, always normal. We don't get shown her blood results here because apparently knowledge is too much power to the patient. So we generally just get told are normal or not. But yeah, so lots of protection. So yeah, our whole family is generally vaccinated. We have some extended family who are choosing not to for various reasons. But yeah, and we are in a fortunate position in New Zealand that, aside from the current situation that we have a small outbreak, we generally get on with life day to day and things. But yeah, if in the instance, is there sort of any data with DC kids, patients with contracting COVID? Or would we, I guess often like people just say to me, oh, Gabby gets it, would it be really bad? And I'd say, well, I don't really know. Is there an answer?
Yeah, and it's one of those things that that we've wanted to do, and we just honestly haven't had a chance to write the study yet. There are a couple different things that are ongoing. So first of all, to know is that we would need to collect the data. There are a couple different things that are ongoing, is that we collaborated, and I think some people from the Team Telomere community did participate. We collaborated with a medical geneticist named Dr. Beth Kozel, who's here at the Genome Institute. And she was conducting basically just a survey of people with rare diseases and kind of how COVID was affecting their lives. So it started off kind of just, day-to-day life. And then there's been a subsequent questionnaire about the rates and things. And so that's still in data collection mode. I know there are some DC families who have enrolled in that study. So that'll be kind of a hint. The other thing that I know of is there is a study with another collaborator here at the National Institute on allergy. And I always say NIAID, so that's why it's the National Institute of Allergy and Infectious Disease. And there's an investigator here who's actually running a study assessing the immune response of the COVID-19 vaccine in immunocompromised people. And she has, it's only fairly recently started accrual, and they have a specific plan, kind of people get vaccinated at home, and they have send-in blood kits that they can send. And that is open to the DC community as well. and a whole host of other things. So much of it is just still collecting data.
So in summary, we just say she's immunocompromised, and that kind of sums it up for the layman. That
okay. Yeah.
When we're talking to school and things, but
exactly.
Like I say, we are very lucky over here, despite our current lockdown status. We're doing pretty good. So
that's great. No, I keep threatening to move to New Zealand. So
oh, come.
Actually, my daughter wants to do her study abroad there. I said, I don't know if they'll let you in. You're an American. So
right now we're not letting anybody in. So not even if you're an actual Kiwi. So
wow.
But one day, I'm sure you'll all be able to come here one day. I hope so. And we're coming to you one day. I just don't know when that will be. Thank you.
Thanks, Megan. Khalil, do you have another question?
I do. Just one last question about the virus on my end. Dr. Savage, as a pediatrician, have you heard, seen, or read anything that indicates how the variant, Delta variant in particular, that we're dealing with now, particularly in Florida? I think Jennifer might be in Florida as well, but I think Jen mentioned, and obviously Megan as well, we all have school-age children. Is there still any indication that this proliferates sort of more moderately between children, or should we be treating Delta differently? And in particular, we notice here in Florida, there's been a big spike in the pediatric wards of hospitalizations. I don't know if that's a function of higher spread in general in the community, or if it's something particular to the kids.
Mm-hmm. So that's a good question, and it's one of those questions that's really hard to answer because it's so many factors. One is that there's simply so much more virus in the community in certain areas that, of course, there are more people exposed. You're going to get more kids who get it. I don't think there's enough data about kind of, are kids more susceptible? I don't think we can say that. We do still know that, you hear a lot of these horrible stories, but for the most part, children are doing okay with it. And but it's also something we want to avoid. So it's one of those things. I think that the data are still coming out, and it's hard in general, in pediatrics, the little kids, the toddler kind of ages, actually aren't very good at, even though we all think, because when you send your kid to daycare, you swear like 2 days later they're sick because they're in daycare, right? But actually little kids don't have a great cough. And so they actually don't spread the virus as far. They don't spread their respiratory droplets as far as older kids or adults. Their cough might sound awful, but it actually doesn't go as far. So, maybe that's a little bit of a saving grace for the youngers, but it's not for for elementary and middle school. It's still, the kids can still transmit it to each other. But I like to think, in fact, this is where I think that different communities, I hope will change and learn, and that, wearing a mask is not a big deal. Our kids learn how to put on a shirt before they go outside. They know they have to wear shoes if they're gonna, go to the store or whatever, and they don't care. They just do it. And I think honestly, a lot of it is the adults who just, I don't know, just don't get it. So I'm hopeful, the kids will persevere, wear their masks. I can tell you granted I'm in Maryland, but my son started high school. There are about over 2,000 students in the school. And he said he rarely sees a person without who's not wearing a mask properly. And this is high school, and they can do it.
I wanted to, Jen, we'll take one more question, but I just wanted to share, and not that our perspective is 4 out of the 5 that live in the house are vaccinated. We did have a breakthrough. Olivia, my daughter, contracted COVID-19. She's a swim instructor to little kids, and we're pretty certain that we don't know exactly where it came from. Her illness time was about 7 days, and it was terrifying to see a positive result in my home with an 8-year-old who's not vaccinated. But it was mild, it was quick, and it didn't spread. So I just wanted to reiterate from my personal experience where we have lived, that small, rare breakthrough, and it was a successful COVID experience for our family. And of course, we're heading into high school where there's no masks out of, the 200 plus people we were in line with the registration. And that's just the hand we're dealt, but we are doing everything we can to keep ourselves safe, and it worked. It did work. So Jen, you had a question?
This may not be a quick question, so if it's not, and I know time might be up, just tell me to talk to you later. But in talking with one of the boys today, he's doing his own research on the vaccine, his concern that he voiced to me, which of course I'm not a science expert, was the fact that the vaccine used the spike protein. And I know you talked about that today. Mm-hmm. And one of the articles that he read talked about the spike protein that's used in this vaccine attaches to the organs, to some organs, and it's causing some organ damage. Is that a myth? Because it sounded like to me in what you told me that it's not possible to do. But is that a quick question that you can answer, or not so much?
No, so it is a quick question, because I think there are other vaccines that are also made that also are to spike proteins and other things in other viruses. So I don't think that it's an issue. I don't think there are enough data. There's not a reason that just this little teeny tiny, it's not like your body's making that spike that you see on that little prickly looking virus. It's making it just enough of that protein so that you can mount an immune response. It is true that the virus itself can cause severe organ damage in really sick patients, but from the vaccine perspective, I would say that's not an issue.
Thank you.
Okay, guys, we're right at 5:07, so we've gone just a little over an hour. I appreciate everyone joining us. Great questions, great conversation. Dr. Savage, of course, thank you always for all you do for the community, and I'm just so grateful that we can share this. This will be available in the next couple of days on all our social media sites and website for you to share if you have families or friends that have questions, or colleagues. We're just grateful to be able to do this, and we'll do a 6-month follow-up and see where we all are in 6 months. But I wish you all health and wellness, and we'll be in touch. Thank you.
All right, thanks everyone. We'll see you all later.
All right, bye.